Okay, so part 3, Contig 25. So what I forgot to say before was that when you click Show Results in New Window, that means you don't have to go back and refresh your search every time. So I am getting impatient, and so I decided to start with the first exon. And so I'm going to do that, and then I'll just start filling these in. I also want to mention, I skipped ahead to the second exon, which is tiny. This is a tiny little exon. It's only 6 amino acids long. And so you definitely don't want to start out with something this short. But once you kind of feel like you have an idea of where you are in the contig, then that's okay. The other thing that I want to mention is that it's useful to take screenshots along the way so that you have kind of a record and you're keeping track of where you're at. So, for example, I should have been taking screenshots all along, but things like, okay, so here we've got a result. These are our coordinates. So I'm going to take a screenshot, and I am gonna open, maybe if I can, I'm gonna open up Word doc and throw that in there, and I may annotate it and say, okay, so I'll move this down so I can write above it, exon 2, or I'll say here's what our gene. Our gene is BT. And then, and in theory, I would also have exon 1 there and put them in order. And obviously, you can trim them down and whatnot, so they're not so obnoxiously huge. Though, frankly, it's kind of useful to have a lot of that information there. So we can have this score, and we'll keep track of what everything looks like and whatnot. And so that way, when you go back, and then when you're actually putting together your posters, you'll have some images that you've saved that can show, problem areas. You're not going to want to show every single screenshot, but if there was an area that was contentious where it was really different from melanogaster, you'll want to actually say, this is why it doesn't quite land up here because of this. And so then you can put up a screenshot and actually see what it looks like. Okay, so now I'm going to blast a few more exons, just maybe the first few in this area, and then we can start annotating at least from there so you can get a sense for what this whole process would look like. All right, so I have filled in coordinates for a few of the first, 5 exons. All right, so now I'm going to go back here, and I don't know why this is down. Okay, I'm going to go back here and just take a look at my isoforms. So here are my isoforms, and I have done the first 5 exons, but also keep in mind that this first isoform is only between 1 and 2 and 3 and 4. And so I just want to, I will start with this one. And so when I figure out the exons for this, I'm going to be keeping track of the phase between my donor and acceptor sites, and make sure that they match no matter what, whether it's this isoform that's between the first and the third exon, where the exon is, or where the intron is, or whether it's between the intron between the first and second. In both of these cases, the phases have to match. All right, so what we're going to do is we're going to go back to the Genome Browser, and I'm going to zoom out a bit till I get to here, and I'm going to focus on this first exon. All right, so in just scanning this stuff up here, I can see immediately that in this first exon, I'm in the second reading frame, because here's my start codon, and I've got a nice long open reading frame across the length of the exon here. So I'm going to zoom in on here and get a beat of my start codon, and it's 3491. So I am going to go and check, and this one's right. All right, and then so those match, so I don't have to do anything there. I'm going to zoom back out till I get to the end of it, and I'll zoom in on here. All right, so now we have a couple possibilities because we've got predicted some some isoforms, the exons might stop here, and with some isoforms, the exon stops here. So I'm just gonna zoom in on this a little bit more. All right, so we are looking, remember that we're looking for a GT, and then in the donor site and an AG in the acceptor site. All right, so where's our AG? We need to kind of scroll. So here's a possible AG, and there's also a possible AG here. So let's just take a look. There's a couple possible ones. So let's just take a look at what we think it is. 3802. 3802 is here. Let me just see. AG, there doesn't seem to usually be what we call a non-canonical splice site, which is one that's kind of rare. But okay, so let's think about this. So we think that it should end, we're predicting that it ends at 3803. Wait, is that right? 3802. We predict that it ends at 3802. That matches with here, but it doesn't match. Oh, you know what? I'm an idiot. All right, we're not looking at the AG. We're looking for a GT. That makes way more sense because GT matches up with where the gene predictions end, and it also matches up with where the RNA sequence. So we're going to go with that this is the base. So we're still 3803, and that, I believe, is still, ooh, we're just one off. So I'm going to fix this on here. And if you want to, you can, when you initially, enter your coordinates, you can make them like a different color so that you know that you still need to go back and check those, especially when you have 43 exons to keep track of. That might be useful. So now I know, okay, we're good. Now here, I'm going to look at what phase the donor, and actually, this is our donor site, and then our acceptor site is at the other end. And so for this exon, I'm actually going to switch these. This is going to make sense in a minute. And then I'm going to do donor phase. All right, so with this one, we have a donor over here. And this is our endpoint. We're in the second frame, second reading frame. So we have a phase of 1 here because we have 1 nucleotide before the cutoff, before the intron. So here's our donor phase. Then our acceptor phase for the next one. So let's go to we'll zoom out and go to our next intron, which this is isoform F that we're doing. So let's look at F. F is a 2. Okay. Whoops. So this is our exon 2, and it's much farther away. And our BLAST results tell us that this starts at 4342. Here is 4342, and now we're looking for an AG. Our gene predictions, I'm going to zoom in just a little bit more. Our gene predictions and our RNA-seq data line up with 4340, and there happens to be an AG there, which is what we would expect. So I'm going to call this 4340, And I'm going to make that black so I know that's been checked. But we also need to check our, our frame here was, so now we should be in the first frame. And so here's where we ended, and now here are phases 2, conveniently, because that matches. So we're going to put this here. So here's our acceptor phase. So basically, I switched this around because for each exon, I will note what the donor phase is at this end and what the acceptor phase is at this end, and I will put those in. And we want this guy to match up with that guy, except I wrote the wrong thing. That's actually 2. And it does, conveniently. So you'll want these 2 to match up, and you'll want these 2 to match up, and these 2 as you go along. All right, so that's basically how you go through and do it. And then I will probably end up posting something as we get further along about how to make the gene model checker work.